Okay, last video of the unit, last topic of the unit, but we'll probably spend a good bit of time with some practice on this. It is linkage and gene distance. So, the concept of linked genes. The concept was kind of discovered and pioneered by Thomas Hunt Morgan, who studied fruit flies, Drosophila melanogaster. Well, Drosophila melanogaster. Okay, so it does. Sorry, I was testing my subtitles to see if they could get Latin names. But essentially the idea of linked genes is that when he was breeding his flies, he was looking at two different traits, body type and wing type. And when he was doing these crosses of his dihybrids, what he found was that he, instead of getting the 933, or not even the 9331 distribution, since it's not a dihybrid cross, he's crossing with a double mutant, he was supposed to get just 25% of each type, but he found that he really only got two types of fly. The wild type, which is the same phenotypes for the two traits as one of the parents in the parent generation, and then the black vestigial, which was the other parental type. So there was no mixing of the two parental genes. And what he eventually determined is that this was likely because the two genes that he was looking at for body color and wing type happened to be on the same chromosome, and so they would tend to be inherited together. Since our chromosomes have a bunch of genes on them, if we are looking at inheritance patterns of specific genes, then we would be seeing that genes on the same chromosome tend to inherit together, because that's what's actually moving and separating during meiosis. We're not splitting up those chromosomes unless crossing over is occurring. And if we look closer at some of his results here, probably not his actual results, but something like it. It wasn't actually 1100. It was 11, and then just a little bit of the two other types. So there was a little bit of mixing, just not nearly as much as he would have expected. It was largely the two parental types that showed up in the second generation offspring. So, to explore more of this idea of linkage, we can return to the pea plants. Genes will be linked together if they're on the same chromosome. So, for example, we could have the color of pea plant seeds as well as if they're wrinkled or not. And we can split them up into two different types, the parental type and the recombinant type. The parental type is the phenotype combination that we saw in the parents, and the recombinant type is a phenotype combination that we didn't see in either parent, but is rather a mix of the two. It's recombining genes from the two parents. So why do we get any recombinant offspring at all? That's where crossing over comes in. Remember when they actually physically lay over each other, undergo synapsis, and exchange genetic material between the homologous chromosomes. And that is what actually will be able to create your recombinant offspring, is creating those chromosomes that do not exist elsewhere in you, that are a mix of your mother and your father's genes. So we are going to, in this last topic, attempt to kind of quantify the amount of linkage that these traits have. And so the idea is that for every 1% recombination, for every 1% of the recombinant offspring that we see, so it is equivalent to one map unit of distance, which is just a made-up unit to indicate how far apart two genes are on a chromosome relative to each other. Because you can imagine that chromosome or genes that are very far apart on chromosomes, if one crosses over, it's unlikely that the other one crosses over as well. But if the two genes are extremely close on the same chromosome, if they're right next to each other, then when a crossing over event happens that gets one, it's likely to get both of them, and so you still wouldn't have that recombinant type. This is showing how crossing over actually leads to the recombinant types. Remember, gray normal is the parental type. They are both dominant traits. And so whenever we see gray and normal, that's one of our parental types. The other is black vestigial wings, which is the other parental type. So we should end up with a 50% parental type chromosomes, 50% recombinant. That's a mix of the two. But in reality, we will not actually see that. So how do we calculate this? How do we actually put a number to it? So the first thing you need to do is identify what the parental and recombinant genotypes are. So you need to look at the parents. You need to say, okay, the parents have this phenotype, and just identify that in the offspring. So in this case, the parental types are gray normal and black vestigial, leaving the other two being our recombinants. And then you need to add up all your recombinants, so just 206 plus 185. You find the total number of offspring, divide the number of recombinants by the total, and write it as a percentage. So in this case, we had 391 recombinants for 2,300 total offspring, leading to a 17% recombination frequency, which, if you remember from 2 slides ago, means they are 17 map units apart. The more recombinants you have, the further apart they are. It kind of falls off at 50%, and 50 map units is kind of as far as they can really be, before just essentially being considered on different chromosomes. But pretty straightforward. The hardest part is identifying the parental and recombinant genotypes, but that is really all you have to do. And remember that the percentage equates to a distance on the chromosomes. Last video of the unit, we'll do practice in class. I'll go over it more in class for now, but that is all. I'll see you later.